Good evening and hello from Davis, California. I wish I was able to come to the UK, but times being as they are, we'll have to do this from my house. So I'm going to talk today about genome editing approaches to augment cattle breeding programs. And I want to set a little background in terms of, I think, the real importance of animal breeding for the overall sustainability of our food production systems. And obviously, breeders can make incredible changes to our animal populations just based on selection for a particular appearance or phenotype. And we've made dramatic changes to our ancestor, the wolf here. And thank goodness we did. I doubt you leave your kids at home with a wolf, but probably quite happy for them to be home with a Labrador. But when it comes to our food animals, we really have made dramatic changes. And I think it's really interesting to look here over the course of only, 40 years? We went from very, very small Angus and Herefords in this case to very, very large Angus and Herefords. And I'm not 100% sure that either of those sizes were economically optimal, but obviously we can make dramatic changes. And ideally, we're making those changes in a direction that's optimal rather than perhaps maximum, which might be evidenced here in the 1980s. I think after that, we kind of got a little bit more economically minded or optimally minded in beef cattle production. And I think that's really nicely reflected in this graph here. And this is a really interesting graph. It shows US animal numbers, that's up against the left axis here in hundreds of or millions. And what we can see is over time, we started off at about 97 million head back in 1960s, and we saw this tick up of numbers until around about 1975. And since 1975, we've seen the population of cattle in the United States go down until it's back to about 92 million head at the current time. But what's interesting is juxtaposed against this on the right axis is meat production, beef production. And we can see that it rose in the early 60s and 70s in line with animal numbers. But then something really interesting happened here where we started to have less animal numbers and more meat production. And there's really only one way that can happen, and that is we're getting more meat per animal. And that has these really big impacts so that today we have about the same number of cattle that we had back in the 1960s in the US, about 92 million head, producing 12.7 million tons of meat versus 7.4 back in the 60s. And a lot of that is due to genetic improvements. And this has a real impact on the overall sustainability of beef production. I think if we compare here, the United States is at the current time at about 90 million cattle, producing, as I mentioned, about 13 million metric tons of beef. Compare that to the next largest beef producer, and that's Brazil, who have 250 million head of cattle, producing only around about 10 million metric tons. And so you can kind of do the math on that and see that there's a really big advantage to having more productive animals. And that's really one of the goals of animal breeding. And so in order to discuss genome editing, I need to go a little bit over the central dogma of biology. And just in case you slept through your biology classes, I'll just remind you that every cell of an organism contains the genome, and the genome, of course, is made up of the DNA double-stranded helix, which is composed of these single nucleotides, the A's and T's and C's and G's. And these are arranged into genes, which make proteins. And you can have small alterations in these nucleotides, which are called single nucleotide polymorphisms. And that leads to alterations or differences between individuals. And the bovine genome is about 3 billion base pairs. And interestingly, if you actually look at the comparison between human and cattle genomes, we're about 83% identical. So we only differ between cows on 17% of our DNA. So if you want to think about it, you're kind of a transgenic cow. And I think as we've got more information about the genome of cattle, we've seen that there's tremendous genetic variation amongst all the different breeds of cattle we have. And there's this one project called the 1,000 Bull Genome Project, which is a bit of a misnomer because it's the 2,703 bovine genome project. And what they found is if you compare amongst all the different breeds, there's about 86.5 million genomic variations or alterations. Those are the SNPs and small insertions and deletions between the different breeds of cattle, which gives them all the remarkable adaptability traits that allow them to live in all of the different environments of the world that they live in. And you can have a thing as simple as a SNP giving you very dramatic alterations in appearance. And this is a very well-known example of the Belgian blue breed, where a single gene has been turned off, and that gene's the myostatin gene, and myo is muscle, statin is stop. So if you turn off the muscle stop gene, you get the muscle go gene, and you get this very large hypertrophied phenotype that is characteristic of the Belgian blue breed. And breeders have been able to use this genetic variation to develop the SNP chips that you're probably all familiar with. And that was basically a technology that's been around about a decade now, and was really the sequencing of the bovine genome that allowed us to develop these chips, which actually enables us to, at one DNA sample, have a look at about 50,000 different genetic variations throughout the genome of an animal, and basically do a genetic merit estimate of that animal based on which SNPs and genetic variants it inherited from its parents. And that's now been adopted globally in dairy cattle breeding programs. And you're probably familiar with the fact that you can get genomically enhanced breeding values that have been enabled by these SNP chips. And it's been rapidly adopted. And what you can see is that since that technology has been adopted by the dairy industry, which happened in about 2009, the rate of genetic gain per year has basically doubled before and after the introduction of genomic selection, because it enabled selection of younger animals, because you could basically take a DNA sample at birth and get a very accurate estimate of the genetic merit of young bulls, and you could start using them as soon as they were sexually mature, rather than having to wait for them to go through a progeny testing program, which typically takes around 5 years. And so this technology has been adopted really rapidly. It's doubled the rate of genetic gain, with almost no public concern or any outcry by groups that are opposed to it. It's just been adopted in the same way that other technologies, such as artificial insemination, are now routinely used in animal breeding programs. So now I'm going to move over to genome editing and look at what that technology is and how it might be used in cattle breeding programs. And so genome editing basically is a very fancy word for a pair of scissors. And so these scissors have different names. There's zinc fingers and talons and very famously CRISPR, Cas9. But what they do is they introduce a double-stranded break. They cut the DNA at a particular location in the 3 billion base pairs that make up the genome. And that location is targeted. In other words, you tell the scissors where to cut. And when they cut, there's two outcomes that can occur. You can either repair it down what I call the left-hand pathway here, where you basically introduce the break and then it gets repaired and it makes small errors, little deletions, little insertions, which actually inactivate the gene, so it no longer produces the protein. So it's a knockout of that gene. The gene is no longer expressed. And you might want that for genes that have a product that perhaps makes your animal susceptible to a disease or produces an allergen in your end product. So there are reasons you might want a gene turned off. The other approach is the right-hand pathway here, where there's a donor template involved in the repair. And basically, when that double-stranded break repairs, it introduces this light blue DNA here, and you get a precise insertion or modification, also known as a knock-in, which can be as simple as a single base pair change, or perhaps you move an allele from Angus into Holstein, or perhaps you move an allele from even a different species into cattle. So depending on what that repair template is, you can get different outcomes here. So I put together a little descriptor that helps to discuss this, and I'm going to display that now.
For centuries, animal breeders have selected individuals displaying desirable qualities and bred them to produce particular goals. breeders are actually selecting on naturally occurring DNA sequence variations that result in differences between individuals, like the color of a cow's coat. Using conventional breeding programs, achieving desired changes can take decades, especially in large livestock species like cattle, which have long generation intervals. Innovations in biotechnology are poised to help breeders improve the rate of genetic gain. University of California, Davis, animal geneticist Dr. Alison Van Eenennaam and members of her laboratory are exploring genome editing tools that could allow breeders to precisely introduce useful genetic variations into livestock breeding programs. Genomes are made up of DNA, containing the collection of genes, unique instructions detailing every characteristic of an individual, what they look like, the amount of milk produced, or resistance to disease. Genome editing offers an opportunity to edit these instructions precisely. One method to edit genomes is the CRISPR-Cas9 system. This system has two components: the Cas9 protein, which cuts DNA, and a guide molecule that directs Cas9 where to cut. Bound together, they form a complex that introduces a precise cut in the DNA. The break is then mended by the cell's natural DNA repair machinery. This can be used to inactivate undesired genes, like those that cause disease. Alternatively, the repair machinery can introduce useful DNA sequences which result in desirable traits, like heat tolerance. Genome editing complements traditional breeding methods. It has the potential to introduce useful traits like disease resistance and adaptability into genetic improvement programs, addressing some of the problems facing global agriculture.
So hopefully that little video helped explain what genome editing is. So let's have a look at some of the applications that researchers have used it for. To be clear, nothing is on the market yet, and it hasn't been commercialized at the current time. And so there are no genome edited animals on the market. But these are some of the research applications. And I've kind of divided the targets into different colors here. And the gene itself is probably irrelevant. It's really, I think, for your interest, the targeted trait, or what are breeders trying to do with this? And so there have been examples to increase yield, and I'll give you some images and later on after we've gone through these. So increased yield, some traits that address animal welfare traits, like introducing polled or not growing horns into dairy cattle genomes, introducing heat tolerance to help animals adapt to a warming environment, the removal of allergens from milk, or improvement of the overall tenderness of milk, or the increased omega-3 product quality in here. And then quite a big effort in the disease resistance. And so there's a number of different disease targets that have been gone after. So eliminating the prion protein associated with mad cow disease, mastitis, bovine respiratory disease, and tuberculosis. And then I'll spend some time on these at the end, and these are applications that would kind of alter breeding program design. So these are all kind of single gene traits, and so let's have a look at some of the research that's been done. This is introducing a gene to fight tuberculosis, and I have a graph up here of relevance to this audience, and I don't probably need to tell you guys that TB has been spreading in the UK, and it seems like it's getting spread by badgers, as I understand it. And so here's an approach to produce cattle that are actually resistant to TB. And I guess I know I'm an animal breeder, but to me, genetic improvement is permanent and cumulative, and it's a better solution to animal disease than having sick animals that then have to be treated with antibiotics and chemicals, or even worse in the case of mad cow disease, where you, or foot and mouth disease, where you had, have slaughtered the animals. And so I think there's a lot of really hopeful applications of disease resistance. Here's a group in New Zealand that knocked out the beta-lactoglobulin gene, and beta-lactoglobulin is one of the major allergens in milk, and so these animals will no longer produce that protein, so that's kind of going down the left-hand side pathway, knocking out a gene. And then here is a group at Texas A&M with two animals. one of which has a knockout of the myostatin gene, and that should ring a bell because I went through it earlier. And just to refresh your memory, this was done using genome editing, and so these are actually Nelore cattle. These are a Brazilian breed, and this one is knocked out and this one isn't. And you can compare that to the naturally occurring knockout of the myostatin gene from Belgian blue. And of course, you can see this animal is a lot more muscly, and so you can get different severities of knockout. And I think that one of the beauties of genome editing is you can be a little bit more nuanced in how you do a knockout, so that you perhaps get an animal that has increased yield, but doesn't have the dystocia or carving difficulty problems associated with the Belgian blue, where they often need to be delivered by cesarean section. And so I think there's a way to tailor the alterations to be a little bit more nuanced than what you are in conventional breeding, where this was probably selected by breeders by just selecting from more muscly looking animals, and that kind of brought that characteristic into the breed. And I'm going to talk a little bit about these animals because this is actually some work that I was involved with. That's me there. And these were animals that were developed by a company called Recombinetics, who introduced the allele that doesn't grow horns from basically the Angus breed into dairy cattle genetics. And so these two bulls are homozygous for the allele that doesn't grow horns. And the way that this was done was, if you have a look at dairy cattle breeds, this is a gene called POLD, and basically the recessive condition is growing horns, and there's a 10 base pair sequence here that in the case of Angus and other Celtic beef cattle breeds, that 10 base pair fragment is naturally replaced by this 212 base pair fragment. And what that does is it makes a dominant allele that doesn't grow horns. And so any breeder knows if you cross an Angus over the top of a horned animal, you'll always get polled offspring because that dominant allele will get passed on from the Angus. And so these bulls were actually brought to UC Davis, and we used them to cross with our horned Hereford. So again, horns is recessive. So horned Hereford cows with this bull produced these offspring. We had 6 offspring altogether. And lo and behold, Mendel knew what he was talking about. They all didn't grow horns. And so that was quite exciting. And we had quite a lot of interest in these animals because they were the first offspring of a genome edited animal. And we had 5 steers and 1 heifer that was born. And she was quite popular with the media. And so we had this cover shoot for her for a magazine called Wired, and they wanted to have her on the front cover. And so they asked if we had a green room we could take photos in, and we're like, well, not really, but we have a shoot. And so they said they'd like to do a cover shot, and we brought her in and she was looking a little rough. And so we cleaned her up for the shot so that you can see her there all ready for her glamour shot. And here's the film crew from Wired. We had a photographer and a photographer's assistant and a director and a director's assistant, all to take a photo of a cow, much to the amusement of the people working out at the feedlot. But there you can see her featured on the cover of Wired. I was a little bit distressed when I saw that picture because she looks a little bit sinister there. And, you know, this is what she actually looked like on the day. So she's not as sinister looking as what the magazine makes her out to be. But then I actually looked at the picture they took of me that day, and I kind of looked like the archangel of death. So I think often the media try to make this technology a little bit more kind of scary than it actually is. So I've gone over a couple of examples of basically single gene traits for disease resistance and improved product quality and welfare. I want to just focus on these two because they're kind of areas of interest for my lab, and that's how you might use this technology to actually alter breeding program design. And so what do I mean by that? So the first one I'm going to talk about is this intra-species SRY translocation. And this is work from a PhD student out of my lab who basically used genome editing to knock in, so right-hand pathway, knock in the SRY gene. And SRY is a gene that's normally on the Y chromosome, and you're well familiar that Y chromosome in mammals makes you a male. And that is the gene that basically triggers down the male differentiation pathway. what we did is we copied that gene onto a chromosome that's not a sex chromosome. It's called an autosome, so the rest of the genome. And then what we predict is this little bull, and this is the genome edited knock-in SRY bull, whose name is Cosmo, is going to produce basically 4 types of semen. So he's going to have Y chromosomes in half his semen, and that when he's crossed with a female, so there's the X chromosome from the female and her normal chromosomal complement, you'll get male because Y, SRY, will give you a male. But then this SRY on the chromosome 17 will also get inherited here, but that will just be a fertile male, kind of exactly the same as this guy actually genotypically. You'll get Y with the chromosome 17 that doesn't have the SRY because he's a heterozygote, so that will also produce a male. But this is where it gets interesting. So he will also produce, half his semen will have the X chromosome, but half of those will also have the SRY on the chromosome 17. Now, we hypothesize that this XX individual will appear male and will grow as a male. It will be infertile because there are other genes on the Y chromosome required for fertility, but it will give you 75% offspring. So you've got 25% for replacement heifers, and all of your sale or market animals will be male. And the male is the preferred sex for basically market animals. They grow more efficiently and have better feed conversion. So that's one thing that could be done. If you're interested, there's a really nice video that Joey made on YouTube that describes in detail how we did that. Another thing that I think is really interesting for extensive industries like sheep and beef cattle production is this surrogate sire technology. And basically the idea here is you would like genetics from this male, but you're going to use this male to deliver it. And so this male is going to be genome edited to basically not have any germline. So he won't have any sperm producing cells. And you're going to basically transplant the genes, the surrogate sire, the genes from this male into this animal, so that when he is then mated with this female, offspring will be the progeny of this male and this female. And so what you could envision and kind of how this might fit into beef cattle production, and I'll use the example here from Australia, where in Northern Australia, you need tropical genetics to be heat tolerant and tick resistant, but they're not necessarily the animals that you would like to sell, especially to the high value export market. There you'd perhaps like an F1, but it's a bit rough on the Angus bulls up there to cope in the hot, ticky environment that is is Queensland. So what the idea here is, normally you have a tropical sire mated with a tropical cow and you'll get a tropical animal, which is slow to mature, it doesn't have the best beef quality. What you'd really like is an F1 that is a combination of this tropical female and Angus genetics, but the Angus male doesn't do well in the tropical environment. So if you create a surrogate sire whose external somatic cells are tropically adapted male, but he's got, if you will, a genetic backpack of his testicles are derived from an Angus, so that when this animal is mated to this cow, you'll get an F1 that's tropically adapted and then also is efficient at beef production and actually has good beef quality. And if you think of it, it's kind of like making artificial insemination on legs. So rather than having to synchronize the cows, you put a bull out, and he does that much better at catching who's on heat and getting them bred than an AI technician can. And they can do it in extensive conditions like Northern Australia. And so I think that's a really exciting potential approach to increase the use of elite male genetics in beef cattle production systems, basically using natural service. And so that's certainly a really interesting area. And so I see editing as kind of the cherry on top of the breeding sundae, if you will. And so breeding programs, they start typically with an association of like-minded breeders, perhaps a breed association. You develop breeding goals, performance recording, progeny testing, AI, ET, genomic selection. Maybe you do some cloning, at least in the US you might. And all of this is still going to be important. You're still going to have that breeding program in place, but genome editing is really the cherry on top of the breeding sundae. It will enable you to introduce useful alleles without linkage drag. So maybe bring in not growing horns into dairy genetics or disease resistance allele into a breed without altering any of the rest of the characteristics of that breed. And I think that precision is what's really attractive. And you could potentially bring in useful novel genetic variation from other species. And that's where we get into kind of more transgenic or GMO applications. And that really brings me to the final point, and that is, are we actually going to be able to use this breeding method? And of course, there's the whole kind of genetic engineering or GMO debate in the background of all of this. And so I think that what happens from a regulatory perspective is really going to determine whether this technology can be adopted. And so let's have a look at the global approach to regulating genome editing. And so this slide asks, would a gene-edited knockout, so in other words, the left-hand pathway where you're just turning off a gene, you're not introducing any novel DNA, be subject to additional regulations in this country? And it's kind of a crapshoot here. So no for Latin American countries, no for Australia, no for Canada, although it depends what trait you're actually working with, because they have a trait novelty trigger for regulation. But yes, at least a current ruling in the EU is that if you use a technique that wasn't in existence before 2001, then you're going to be regulated as a full-on GMO, even if the edit you make is exactly the same as an existing alteration that exists in cattle, for example, the Belgian blue mutation. So you can absolutely replicate that, but you'll be a GMO because you used genome editing to introduce that deletion. New Zealand is kind of EU in disguise. And then you might be interested to see that at the moment, the US is proposing also to regulate these as new animal drugs, which was the approach they used for genetic engineering. The USDA has recently proposed an alternative to that, and there's a kind of a rulemaking in progress that might make that so that that's not the case, and they would be more aligned with the South American approaches, which is basically, if you could have achieved that edit using conventional breeding, then you're conventional breeding, and we're not even going to regulate you any differently to conventional breeding because we don't regulate conventional breeding. And so that's kind of the approach that the South American countries have taken. What about if you gene edit a knock-in? So in other words, for example, you bring in the 202 base pairs that make cows not grow horns in the case of polled. Would you be subject to additional regulations? And the only alteration here, I don't know if you noticed it, is that in Australia, that would trigger regulations because their trigger is if you use that repair template to basically repair the double-stranded break with the little blue DNA that I showed in my diagram. And so, for example, introducing polled into the Holsteins in Australia would be regulated, but knocking out a gene like myostatin would not be. And of course, the EU is regulating everything irrespective. So you're in an interesting situation with Brexit, and I think that you have the potential to actually get some science-based regulations around this. And so I encourage you to really do that, because I think this technology has a lot of potential to introduce traits like disease resistance. And I guess I'll just finish with my warning or my trade-off concern, and that is, there are real trade-offs associated with saying no to animal breeding innovations. And I'll just draw your attention back to the 1940s when AI, or artificial insemination, was first introduced. It was a little bit controversial. It's since no longer controversial, but I think that the improvement in genetics that was enabled by AI, where you basically had a 4-fold increase in the production per milk cow, and in the US, that resulted in a decrease of the dairy cattle population from about 26 million down to 9 million, and overall had the effect of basically making a glass of milk have only 1/3 of the greenhouse gas emissions today that it had back in the 1940s, because you've got less cows producing more milk. And so you could have said no to AI, but what would be the environmental implications? And I guess my question would be, you can do without the cherry on top of your sundae, but it'll be associated with basically slower rates of genetic improvement and potentially not being able to introduce traits like disease resistance, which come along with their own impacts, which is obviously the animals will still get sick, and that's has environmental and kind of sustainability issue concerns as well. And so I think those considerations are really important in the discussion around this as the UK decides how they're going to regulate this, because the opportunity cost of not using technology should very much be part of the discussion rather than just potential risks of adoption of the technology. And so I thank you for inviting me here, and I look forward to questions at the end of the session. Thank you.